Okay, well, in any case, okay, so I've got 4:15 on my watch, so I'm going to start. So we have 1.5 hours, right? And I hope that I'll be able to get everything done in time because there's actually quite a lot of things that I want to present to you today. So basically what I'm going to do is give you some reminders about basic coronavirus virology and a little bit about, a reminder about the pathology of COVID-19 compared to seasonal human coronaviruses. So that will be the background. And then in terms of the immune response, I'm going to talk a little bit, well, firstly about innate responses, particularly the interferon system, and a little bit about the concept of the cytokine storm. And then afterwards, I'm going to go on to the adaptive response, first T cells and then antibodies. And this will bring me on to the topic of these variant viruses, their escape from neutralization, and I'm going to end with a little bit about neutralizing epitopes. So that's the overall plan. Well, I tried to catch up with a lot of recent publications, so I think it's going to be up to date today. And like I said before, I'm going to try and keep on time, okay? So there generally won't be very many pauses during the lecture, and so I'm just going to keep on going unless there's some like a major technical thing, like you can't hear me or see anything anymore. So in that case, please tell me, but otherwise we might need to take questions at the end, okay? Right, so I'm going to start out with a reminder about coronaviruses. Okay, so as you all know, until a couple of years ago, there were 4 endemic human coronaviruses, 2 alpha coronavirus species and 2 beta coronavirus species, which cause seasonal infections, every year, and we all catch these regularly. And then up until a couple of years ago, there were known to be 2 new beta coronaviruses, the SARS coronavirus, SARS for severe acute respiratory syndrome coronavirus. So this was a very serious lung infection that emerged in the south of China and was transmitted among people from 2002 to 2003 before it was possible to eradicate the virus. MERS is for the Middle East Respiratory Syndrome, so another beta coronavirus, this time transmitted to people from camels, and it's really only causes sporadic infections in Saudi Arabia, Jordan, and countries where people come into contact with the intermediate animal reservoir. And then that brings us to SARS CoV2, which has kind of won the jackpot in terms of in the coronavirus lottery. So this is the one that's brought the world to a halt for the last 18 months. Now, yeah, it's always worth repeating that the virus is called SARS coronavirus 2 and the disease is COVID-19. So it's like HIV/AIDS. Okay, so I'll try and follow this convention throughout the talk. Okay, so coronaviruses, they are enveloped plus-strand RNA viruses with spherical particles about 120 nanometers in diameter. And they are called coronaviruses because the spike glycoprotein trimers form a crown around the outside of the envelope. And you can see that in electron microscopy. So the name of the virus family comes from their morphology. Now, inside this spherical particle, there's a helical nucleocapsid. So the nucleocapsid is like a long filament wound up inside the particle. Now, there are only 4 structural proteins in these particles. Okay, so on the inside, you've got virus RNA complexed with the nucleoprotein. So that's the nucleocapsid. On the outside, you have the spike glycoprotein trimer, and in the membrane or in the virus envelope, you have two integral membrane proteins, the membrane protein M and the small membrane protein E. So basically the functions of these proteins, so S, the spike, is the only protein involved in receptor binding and entry. M and E have functions to do with virion assembly, release, and the shape of the maintaining the spherical shape of the particle. Okay, so quite a simple virus particle, not too many proteins, and the one we're mainly going to be interested in is the spike glycoprotein today. So once this virus infects cells, it's going to start a typical replication cycle of an RNA plus strand virus. So just like for any virus, the replication cycle starts with attachment to a host cell, then entry and release of the genome by decapsidation. Now, since the genome is plus strand RNA, it kind of looks like a messenger RNA, right? So the genomic RNA can be directly translated by host cell ribosomes. So you directly get expression of some viral proteins. Now, amongst these viral proteins, there will be an RNA-dependent RNA polymerase, which can copy the genome into a negative strand copy, and then copy this negative strand intermediate into more copies of the genome. So other viral proteins expressed will be the structural proteins, and these will assemble with the new copies of the virus genome to give you new particles, which are then released. And all of this occurs basically in the cytoplasm. Okay. So thinking about the first step, attachment and entry, we know the receptors for different human coronaviruses. So, the endemic human coronaviruses, I think one of them binds to sialic acid as a receptor, but the NL63 binds to ACE2, 229E binds to CD13, and SARS and SARS coronavirus 2 both use this same ACE2 receptor. So once the virus is bound to the receptor, then it's internalized either by endocytosis, or it will initiate membrane fusion directly after an interaction with a co-receptor. However, in both cases, what is really essential for fusion is partial proteolysis of the S-glycoprotein, and that's what's going to activate membrane fusion. And the membrane fusion will allow the nucleocapsid, that's the virus genome, to be released into the cytoplasm. So to have a little bit more of a closer look on how this works, the spike glycoprotein is expressed as a trimer on the surface of the virus, which you can see here. So there's a different color for each monomer, and each monomer has two domains. The S2 domain, that's these alpha helical region here, and the S1 domain, which is kind of more on the top side of the spike glycoprotein. So you have to imagine the viral envelope membrane is down here. This is the outside of the virus up here. So this C-terminal domain of S1, it's also called the receptor binding domain. So this is what's involved directly in binding to ACE2. And S2 is the fusion domain. So these alpha helices will be, will change conformation and project this fusion peptide in red into the membrane of the host cell. And that's what is going to catalyze membrane fusion. So I'm just going to try and show you this YouTube video here, if it wants to come up. Where is it? Let me just bring it over. Okay, can you see this? Okay, so I'll just play this. So you've got this kind of structural model of a SARS coronavirus 2 particle here. Okay, so nucleocapsid proteins in the middle and spike glycoprotein trimers floating around on the outside. So one of these particles is going to come and bind to the surface of the cell here. And in purple, you have the ACE2 receptor, and you can see the RBD, the receptor binding domain, kind of flips up and allows it to interact with ACE2. So you'll have like sequential interactions with 2 or 3 ACE2 proteins, and then the co-receptor here, which is TMPRSS2, will cleave at the S2' site, and that's what's going to activate the fusion domains here. So after this proteolytic cleavage, then you'll find that these fusion domains are going to unfold and project the fusion peptide into the cell membrane. Now this intermediate form is unstable, so it's going to collapse, and this generates force to bring the virus envelope together with the cell membrane. And then what's going to happen is the virus envelope is going to dissolve in the cell membrane and liberate the nucleocapsid into the cell cytoplasm. Okay, so basically that's how it works. And actually I should have gone through this first. Okay, so in order for this to happen, okay, there are 2 stages of proteolytic cleavage that need to occur. So first, S1 and S2 have to be separated. So that's called priming of the spike glycoprotein. And then this S2 site, S2' site here needs to be cleaved. So that's activation of the fusion domain. Now, in seasonal coronaviruses, the particle enters the cell by endocytosis, and then it's cathepsins in the endolysosome which cleave at both of these sites and activate membrane fusion. However, with SARS coronavirus 2, then the S1, S2 site is cleaved during release of the particle from infected cells. So it's furin which does this. So the spike glycoprotein is already primed. Okay, it just needs one more cleavage to activate fusion. And this can occur at the plasma membrane by the co-receptor TMPRSS2, which will cut directly here and activate fusion. Okay. So it's kind of a slight difference in entry mechanism between SARS CoV2 and the endemic human coronaviruses. So after membrane fusion, the virus genome plus strand RNA gains access to the host cytosol. So if you look at the structure of the genome, it's about 27 to 30 kilobases long. So it's a kind of biggest genome among single-strand RNA viruses. And if you look, you've got several open reading frames. At the 5' end, you have 2 large open reading frames that code for polyproteins. And at the 3' end, you have several short open reading frames. Each one of them is going to code for an individual protein. So the structural proteins are coded here, and the proteins involved in virus replication are coded up here in these 2 large open reading frames. Okay, so one thing that you can notice also is in infected cells, you find full-length genomes, and you'll also find viral messenger RNAs that code for one of these proteins at the 3' end of the virus genome. However, all of these messenger RNAs, they all share the same 5' leader sequence with the virus genome. And we'll see why that's important a little bit later on. So how does this happen? Well, the first thing that happens is, the host cell ribosomes will bind to the 5' end. They are capped, so they're recognized by host cell ribosomes, and translate the PP1A and PP1AB polyproteins. So these polyproteins are then cleaved by the viral proteases into functional proteins. And what is important here is you have an RNA-dependent RNA polymerase. So that's coded in what's called non-structural protein 12, NSP12, and a helicase function, which is also essential for replication. So this RNA-dependent RNA polymerase will form negative strand copies, either full length or shorter ones, which will be the templates for production of virus RNA. So each of these negative strand templates will recopy a part of this end here. So that's how you get the same leader sequence on all the messenger RNAs. Okay, so in the end, you get these messenger RNAs. And of course, this is all happening in the cytosol. So the virus has got to add a 5' cap and a poly A sequence to these RNAs itself. So it has the enzymatic capacity to do this. So you get first these proteins involved in replication expressed, and then later on the structural proteins. And then another thing that's important here is, the coronaviruses favor their own protein expression by shutting down host cell protein synthesis. And that's a function of this NSP1 protein. So NSP1 binds 18S ribosomal RNA at the mRNA entry channel. So, if host mRNAs are bound by the ribosome, they can't actually move forward. So no host proteins can be expressed after coronavirus infection. So how does the virus express its own proteins? Well, that's the role of this 5' leader sequence. So another domain of the NSP1 protein will specifically bind to these secondary structure elements, these stem loop structures in the 5' leader sequence, and that allows the release of NSP1 so that the viral RNAs can be translated. So, this is quite clever. The virus is going to completely take over the translation apparatus, block all the host protein synthesis, so all of the infected cell ribosomes can be dedicated to producing viral proteins. So the last step in the replication is budding and release, and this appears to occur not at the cell surface, but at the ER Golgi intermediate compartment. So here you can see on electron microscopy, this is the endoplasmic reticulum, this is the Golgi network, and coronaviruses bud somewhere kind of into this in between them. Okay, so this ERGIC, and then once the particles are here, they just get released by the normal secretion pathway. Okay, oh, excuse me, that's my dog. All right, so I close the door, he won't come back in. Okay, so that's a basic fundamental virology. Now a little bit about pathology of coronaviruses. Okay, so for seasonal human coronaviruses, the endemic ones, they cause like seasonal respiratory infections. So mostly they are, relatively non-pathogenic, not very serious, but they can cause pneumonia, okay, and in some cases gastroenteritis. So in fact, if you look at children in France who are hospitalized for respiratory infections, about 10% of these children will have been infected by human coronaviruses. So they're not entirely harmless, okay? These guys have some kind of pathogenic potential. Now, there's not so much information about pathology in kind of community transmission because, people aren't really that sick. But I did find one study published a couple of years ago looking at a community in New York. So here they followed children infected either in the community or hospitalized. And here you have the different symptoms, and these two figures here are for the percentage of children in the community or in the hospital who have these symptoms. So a normal infection, you have a blocked nose, a cough, not really much fever, and no wheezing. So wheezing is kind of like a particular kind of sifflement in the lung, that the clinician can hear with the stethoscope. So no real lower respiratory apparatus infection. So hospitalized cases, more fever, more wheezing. So you get this viral pneumonia that causes hospitalization. And I forgot to say that the people who are at risk of a more severe infection with seasonal human coronaviruses are very young children, so no acquired immunity, and then older people and immunosuppressed patients. So these older people with immunosuppression, these are also two risk factors for severe SARS CoV2 infection. So how about SARS CoV2? Okay, so this is a slide from last year, but I think it's still true. So the incubation period is about a week, 6 days perhaps on average. So the incubation period is the time between when you get infected and when you start to show symptoms. 6 days, maybe 1 week. Then you get the first symptoms, which are fever, cough, fatigue, loss of appetite, muscle pain, and diarrhea. And then these symptoms will kind of either attenuate and then you get better over the next week or two, or they might get worse. And then after 1 week, you'll find perhaps some difficulty breathing, and then this becomes more and more severe. And then some patients will need oxygen support, and the worst will need ventilation, mechanical ventilation. So what seems to be happening is you've got like different stages in the disease pathology. So these early symptoms, they really seem to be caused directly by virus replication. So, you know, fever, you've got your innate immune response kicking in, and, you know, as a direct result of high viral replication. And then during this moderate infection towards a more severe infection, then it seems to be mainly due to a runaway host inflammatory immune response. Now, this means that, you know, once you have someone with a very severe infection that's in the ICU, they won't really respond to antivirals. If you're going to use antivirals, like antibodies or small molecule inhibitors, you really need to use them very early in infection. Whereas here, the clinical management is going to be to do with, you know, dampening down this host immune response and trying to help people to survive. Okay, so that's kind of the clinical manifestations of the disease, but, you know, we all know that only a minority of people will have a severe infection and will be killed by this virus. So how do the figures really work out? So one of the best papers I saw on this last year was a group of epidemiologists in the Pasteur Institute. So they analyzed all the data in France from about February to May in 2020, and then broke everything down by age, and tried to get a good estimate of the total number of infections for each age group. So for each age group, they calculated the probability of being hospitalized if infected, and the probability of death if you were hospitalized. And then they managed to get an estimate of this, which is the infection fatality proportion. That is, what is your risk of dying if you get infected? So, you know, for a young adult here, like 20 to 29 years old, and basically this will be, you know, less than 1 in 10,000 chance of dying if you're infected with SARS CoV2, you know, if you're a young, healthy adult. Now, as you get older, you know, the risk gets higher. And that means that with the overall population, risk would be about 0.5%. So, you know, if everybody in the French population had got infected last year, then we could have had, oh, I don't know, what would that be, like 1%? Could it be like 300,000 deaths or something like that? Okay, so that's true. That was true for the wild type virus, but we know that we don't have the same virus now. Now everybody's getting infected with the Delta variant. So are these variants more pathogenic? So one of the studies I found on this was from the UK, looking at the Alpha variant, which is also called B.1.1.7, from 16th of November 2020 to the 5th of February 2021. And here they were able to analyze, you know, 185,000 infections. And they were able to do this because they used a kind of national database that covered about 40% of the whole population. So out of these people, they were able to guess or evaluate which viruses were the alpha variant and which ones were the wild type. So the variant of concern is alpha variant, and non-VOC is wild type. So over this period of time, you had the virus population in the UK really being completely dominated, you know, from first appearance here at the end of October to, you know, March 2021, the virus population completely changed. But over the study period, you basically got about half and half, all right? So they had like 90,000 cases for each virus infection. And then they were able to compare the case fatality ratio between the wild type virus, non-VOC here, and the variant of concern here. So if you look once again, in people under 65 years of age, the wild type case fatality ratio pretty close to 0.1%, a little bit higher with the alpha variant. And the same was also true for older people, so for a woman between 75 and 85 years old, then this would be about 4.7% risk of dying after the infection with a wild type SARS CoV2. With the alpha variant, this risk goes up to about 7.3%. So overall hazard risk of death of the alpha variant compared to wild type was about 1.67. So that means it's about 67% more pathological. So a significantly increased virulence here. So how about Delta? So there's been a similar study published in Canada where they analyzed data from February to June this year. And here they were able to compare about 6,000 infections with Delta variant, 162,000 with the alpha variant, and 43,000 with the wild type. And then here we go for the probable Delta variant. Then they found that the hazard risk of dying was about 2.3 compared to the wild type. So the Delta variant seems to be twice as virulent as the original SARS-CoV-2. And when they compared alpha versus the wild type, they got a hazard ratio of about 1.51. So pretty close results for alpha between the two studies. So I think it is reasonable to say that the Delta variant is about 2 times as pathogenic as the wild type. So significantly more dangerous. So where are we in France now in terms of epidemiology? So these are the numbers of daily average cases, average over 7 days. And these are the number of deaths ever since the beginning of the epidemic here. And one thing you can notice is that here you've got a big peak of deaths and hardly any cases detected. Well, that's because right at the beginning, nobody had testing capacity, so nobody could detect all these cases. And then in this middle part, every time we've got a peak of cases, we've got a peak of deaths. But now we've got like this region here, where we've got a lot of cases, almost no deaths. And that's probably due to vaccination. Okay, so more people are vaccinated, maybe getting infected, but not really dying so much. And now we're going to see what's going to happen in the next couple of weeks with this new peak that's just started out here. Okay, so that's the general introduction. So now I've got about an hour to really talk about the role of the immune response, and we're going to start out with the innate responses. So for the interferon system, now one thing I like to put up on here to basically remind myself is that we talk about interferons, but there are many different interferons, which can be classed into type 1, type 2, and type 3 interferons. So type 1 interferons are mainly interferon alpha, interferon beta, and then these weird ones, epsilon, kappa, and omega. So interferon alphas are mainly secreted by peptitoid dendritic cells. Interferon beta can pretty much be secreted by all nucleated cells after virus infection. Type 2 interferons, interferon gamma, which is expressed by T cells and NK cells, and type 3 interferons are interferon lambda, which is expressed by epithelial cells. So type 1, type 2, and type 3, the classification makes sense because these different interferons, type 1, type 2, and type 3, use different receptors. So the interferon alpha receptor basically works for all of the different type 1 interferons. And they are extremely important in the antiviral response because what will happen after binding to interferon receptors is that you'll get signaling pathway which will result in the expression of a couple of hundred interferon stimulated genes called ISGs or sometimes IFIs for interferon induced. So these ISGs have got direct antiviral effects and render the cells more resistant to virus infection. So how do we know that the interferon response is important in COVID-19? So one of the first bits of data is, take bronchoalveolar lavage cells from patients with a severe SARS CoV2 infection and do RNA sequencing, and then compare the profile with patients either who've got other types of pneumonia, and also compare them against healthy donors. So in severe SARS CoV2, you've got a lot of genes strongly upregulated, and among the upregulated genes, you've got a lot of these ISGs. So we know that there's a strong activation of the interferon system during a SARS CoV2 infection, especially in a severe infection. Now, another indication that the interferon system is important in survival is looking at which genetic variants over the whole genome are associated with severe infection. So this is a GWAS experiment, so genome-wide association study. So basically you get a whole bunch of cases, a whole bunches of controls, and you genotype them for SNPs all over the genome, and you try and find particular alleles or SNPs, that single nucleotide polymorphisms that are associated or that are enriched in the cases. So the cases here are people with severe COVID-19 compared to milder asymptomatic infections. So the first publication here was from a group in Italy and Spain. What's kind of surprising is they only really found 2 loci involved. This is the blood group antigens. That's rather interesting, and kind of strange story attached to that. And then there's this locus on chromosome 3. So here you've got a bunch of chemokine receptors, and you also have this gene LZTFL1, which hasn't been recognized as an antiviral effector or involved in the immune system response at all, but this may be directly involved because it's strongly expressed in the lung epithelium. So I don't think anybody really knows what's the basis of this one. However, another similar type of study was published at the end of 2020 in Nature. So here you have more cases, so the N is a bit bigger. It was done in the UK, and they managed to just compare individuals of European ancestry. So there's a bit less background. So they picked up the same 2 this locus on chromosome 3. Blood group antigens didn't really come out, another locus here. And then amongst the other significantly associated locus polymorphisms associated with severe COVID-19 infection, you have this OAS1 and the interferon alpha receptor 2 gene. So for the interferon alpha receptor 2 polymorphism, the risk allele is associated with lower expression of the receptor. So you don't have so much interferon alpha receptor, so you don't have such a good interferon response. So that makes sense. OAS1, I'll talk about this a little bit later. But anyway, so you have it's an interferon, it's an ISG, okay? So you have one ISG and the interferon alpha receptor associated with severe cases. So this is telling you the interferon system is important. Now, another way of looking at this is not by looking at frequent genetic polymorphisms like in a GWAS experiment, but trying to find rare genetic, rare mutations which are only found or found preferentially in patients with a severe infection. So this was a study published last year with a big international consortium coordinated by Jean-Laurent Casanova. So it included 659 patients with a severe infection, and they sequenced the whole genome or the whole exome, and they looked for mutations in 13 genes involved in the interferon system, either involved in activating plasmacytoid dendritic cells that express type 1 interferons, or involved in detecting the presence of viruses, and or involved in the type 1 interferon signaling pathway. So they detected 118 variants, and 9 of them were complete, deletion, loss of function mutations, and 109 were, missense or inframe deletions. And they functionally tested them, and they found 24 deleterious mutations by, testing their function on PBMC or other in vitro assay systems. So they found that these rare mutations were found in about 3.5% of patients with life-threatening coronavirus, and almost absent from people who had asymptomatic or mild symptoms. So the odds ratio, 8-fold more frequent in people with severe life-threatening disease. So these mutations are quite rare, and they hadn't been reported to be associated with susceptibility to other viral infections. So people didn't have like a past history of recurrent viral infection. So you might have one of these genetic risk factors, but you'll never know about it, okay? Another thing that is also very important, I think this is probably one of the most important papers published last year, was the finding that many of the people with very, very severe COVID-19 infections have autoantibodies against type 1 interferons. So this was a big French consortium who screened patients for antibodies against interferon alpha 2. And they had 987 patients in the case group and 663 controls, either with mild COVID and 1,200 healthy donors. And here are the results for the interferon alpha binding of antibodies in the serum. So, from time to time, you find some people in the controls with these anti-interferon antibodies, but it's much more frequent in the patients with very severe infections. So these antibodies are neutralizing. So here, this is an assay on monocytes from the peripheral blood. And if you add interferon alpha 2 to these monocytes, or interferon omega, then STAT1 is phosphorylated, and that's what you detect here by flow cytometry. So you've got STAT1 phosphorylation activation by interferon. However, if you put in serum from one of these patients, which has got anti-interferon alpha 2 in the binding assay, then you completely block STAT1 phosphorylation. And if you've got a serum that is binds to alpha 2 and omega interferon, it will block both of these interferons. So these antibodies are neutralizing, they block interferon signaling, and there's pretty good relationship between the strength of the binding and the neutralization assay. Okay, so the more of these antibodies you have, the more they will block interferon signaling. So one of the things that was rather worrying is that all of these antibodies tended to cross-react with all the different interferon alphas and a little bit of interferon omega, but not interferon beta. So that means if you want to treat some of these patients with interferons, or you could use interferon beta, but not interferon alpha. So there's a follow-up paper to this one published this year where they looked at several thousand people. Basically, the result is that they confirmed that these anti-interferon antibodies are enriched in patients with critical COVID-19 infections, and that's 9.8 to maybe 14, 10 to 14% of patients. So if you put this together with the rare mutations, then it's probably about 15% of all the critical COVID-19 cases are related to some errors in the interferon system, either these autoantibodies or some mutations that are inherited. So this is a pretty important thing, I think. And of course, the autoantibodies, the older you get, the more you have them. So it kind of explains partially why you get more severe infection in older people. Okay, so moving on a little bit, then, so what really is the effect of interferon? Why does it block virus replication? So I'm showing you this slide just kind of like an example of an experiment in cell culture. So you've got human airway epithelial cells here, and you can infect them with SARS coronavirus 2, and then 12% of them are infected after, I think, 72 hours here. If you pre-treat with interferon beta, you'll have 100-fold less infected cells. So pre-treatment with interferon beta really effectively inhibits virus replication. So which ISGs are doing this? Which ISGs are rendering the cells resistant to SARS CoV2 infection? So there have been a few studies on this. So basically what people do is, the research groups involved, they'll get few hundred ISGs, and then transfect cells, each well will be transfected with one particular ISG. And then they infect the cells with SARS coronavirus 2, and then look at 40 hours later to see how many infected cells you have. So this was published early this year. They screened about 400 ISGs, and they found 65 which had a direct antiviral effect against SARS CoV2. So this was the list of the top scoring genes they had with the biggest antiviral effect. Some of them affecting the entry stages, others affecting the late stages. So this is another study here, similar kind of thing, similar kind of experimental design, but they got a list of different viral proteins involved. Probably they're using a different cellular system. So what's interesting for us here is that they picked up OAS1. So this had already been identified in the GWAS. So I'm just going to talk a little bit about this one because it's, I think, got a really interesting story here. So OAS, what is that? That's for 2', 5' oligoadenylate synthase. Okay, so oligoadenylate, this is a short molecule of RNA. Normal RNA is polymerized, is connected 3' to 5', right? So this is a kind of unusual RNA. So basically OAS, what it does is it will recognize double-stranded viral RNA. When it's activated, it produces 2', 5' oligoadenylate. And then this 2', 5' oligo A activates RNase L. RNase L will degrade all double-stranded RNA, cellular or viral. So it's directly going to destroy the virus genome. Okay, so they had this group here, they're from Glasgow, in fact. They looked at the antiviral effect of OAS1 against SARS-CoV2 and a bunch of other RNA viruses. So its antiviral effect is really pretty specific for coronaviruses. And, so it recognizes double-stranded RNA, but it's also sequence specific. So they mapped the binding sites of OAS1 on the virus genome, which you can see here, and they found a bunch of them in the 5' UTR. So here they found that they had OAS1 binding sites in these stem loop structures in the leader part of the viral mRNAs. So if you remember, these stem loops were essential for virus protein translation because they need to be there to remove NSP1 and unblock the host ribosomes. So this, I think, is really clever. You've got OAS1, which is recognizing a structure which is really essential for virus protein expression in virus mRNA. So it's not really that easy for SARS-CoV2 to change this. Okay, so as we saw, there's a protective allele and there's a risk allele in European populations of OAS1. So what's the difference between them? So an interesting thing in this paper is that, okay, they were able to find out how this is occurring. So the protective allele allows exon 5 and exon 7 to be spliced together, and this will produce a kind of like C-terminal extended form of OAS1. And what it's got here is a prenylation site. So the P46 form of OAS1 will be prenylated, and that's going to direct it towards intracellular membranes. Whereas the risk allele, the P42 form, isn't prenylated, it's just going to float around free in the cytosol. So why is this important? Well, it's important because coronavirus replication occurs in a kind of specialized replication organelle, so these kind of like membrane-bound compartments here that are close to the endoplasmic reticulum. So you can see this in a kind of like cryo-electron tomography. So these kind of yellow double membrane vesicles, this is where the virus replication occurs. So the protective form of OAS1 will be directed towards these membranes. So it's in the right place to detect virus replication, activate RNase L, and destroy the viral RNA. So basically that's how this is working. And the protective allele in European populations, interestingly enough, has got a Neanderthal origin. Not true for African populations. So they have the protective allele, but it's a kind of ancestral form that kind of disappeared from modern humans in Europe. Okay, so next thing, cytokine storm. I'm going to go through this rapidly, I think, because we have about, we've got 45 minutes, that's okay. So the cytokine storm. So one of the initial observations, from early 2020 is that, patients who've got severe forms of COVID-19, they've got, high, increased plasma levels of cytokines and chemokines like IL-1, IL-6. So, this is one study, and then this is another study from 2020. So here they're comparing severe cases with moderate cases, and you can see that things like, IL-10, IL-6 are significantly increased in severe cases compared to moderate cases. And then there was a bit of confirmation in some other studies later on in 2020. Once again, mechanical ventilation, yes, no. So these people have a more severe infection. And you could look at the IL-6 levels here, they're much higher in the patients who then required mechanical ventilation. This was one evaluation cohort. Same association was found in a validation cohort. So, IL-6 was found to be a reasonably good predictor of, more severe infection. So these kind of observations were accumulated, last spring, summer, and then there was a kind of comment article in Science in May 2020, which suggested that people with severe disease, then it looks like this cytokine release syndrome, where you have a very very active inflammation, a cytokine storm, and then they suggested that this is what is causing the pathology. That's very important, because if that's true, then you can probably block the pathology with antibodies against IL-6 or something. So this was the comment in May 2020. I think it was quite influential. So, but other people have tried to look at this idea with a bit critically. So this was a review of meta-analysis published in October last year. So they tried to look at all the available studies and to find out, how much IL-6 have you really got in people with critical COVID-19. And look at this here, not really that high, okay, compared to all COVID-19, or the severe and the critical cases are not really massive, especially if you look at the real acute respiratory distress syndrome, where you've got massive levels of IL-6, or this cytokine release syndrome, where you really have much higher levels. And compared to sepsis, severe COVID-19, it's not really that high. So, that's one indication that this is maybe not so important. And then, it's another study later on in 2020. They looked at multiple cytokine measurements in COVID-19 patients, with a quite a high death rate, and to see whether they've got really significantly higher IL-6 or IL-17A in these critical COVID-19 patients compared to patients with, influenza, pneumonia, or this cytokine release syndrome. So once again, you've got, these cytokines are elevated compared to the healthy controls, but they're not like massively increased compared to people with pneumonia from influenza. And it's certainly much lower than what you see in these syndromes where, you have a really very, very strong release of IL-6 and other pro-inflammatory cytokines. So the conclusion of this article was that, the cytokine storm phenotype, you can find it, but it's, 3 to 4% of patients, and it's not really the thing that explains all of these critical COVID-19 cases. So, that's really the conclusion of this. So cytokine storm, it exists, but it's, it's a subgroup of patients. It's not all of the critical patients. 3 to 4%, I think, is a reasonable estimate. Okay, so now I'm going to switch my focus here. I'm going to talk about the adaptive responses, first T cells and then antibodies. So the first thing you need to know about T cells is that T cell lymphopenia is a major manifestation of severe COVID-19. So it's one of the first biological markers of severe COVID to be identified. So you look at the lymphocyte count here in patients who are in the ICU. It's much lower than patients who don't need intensive care. And conversely, there's an increased neutrophil count. So if you've got a high neutrophil to lymphocyte ratio, this is always associated with more severe COVID-19. So basically, you've got fewer T cells in the peripheral blood. And this seems to be a combination of migration to the inflamed lung tissues and also perhaps a bit more of a kind of activated and activation-induced cell death phenotype. So there's some elimination of T cells and some migration. It's a combination of these two things. So now, is the T cell response significantly different in patients with mild cases and severe cases? So this was studied, in a number of very good papers last year. So I'm just going to present one from people at the Karolinska Institute in Sweden. So here, all the time, they are comparing. Okay, blue, this is healthy donors from 2020. Green, these are patients who have recovered from a mild infection, so they're convalescent. And then in yellow, people who are symptomatic but with mild symptoms. Red, symptomatic with severe symptoms. Okay, so here they're quantifying the number of activated CD38 positive, PD1 positive CD8 T cells, or CD38 positive HLA-DR plus cells. So you've got more activated T cells in the symptomatic patients, but no real difference between critical cases and mild cases. So one of the things that they did was that they I looked at the specificity of these cells. So most of these activated cells here seem to be specific for the virus. So most of these are virus-specific T cells. So that means you've got pretty strong, robust T cell response, both in mild and in severe cases. And when they looked over time, okay, then at the virus-specific T cells, now as people go longer without symptoms, then their effector functions are reduced, and then these CD8 cells get more of a kind of memory phenotype expressing CCR7, but also CD45RA. Okay, so these SARS CoV2 specific CTL get a more central memory phenotype as people recover. So okay, so there are CTL that are specific for the virus around, but maybe their function is different. So here they're looking at interferon gamma, IL-2, TNF secretion from virus-specific T cells in patients who have either mild infection, so that's the kind of like brown bar here, and people with severe infection, that's the green bar. So basically not much of a difference, okay, people with a severe COVID or mild COVID. So no obvious functional difference. Okay, so that's at the acute phase. How do T cells persist after the acute phase? So this is a paper from Shane Crotty's group in California. So here they are stimulating CD8 and CD4 T cells with pools of peptides from these different viral proteins, and they are quantifying how many activated T cells you have after this stimulation. And then they put all the data together with days post-symptom onset. So you can see that the CD8 cell response seems to decline over 6 months after infection, and the same for the CD4 T cell response. And phenotype kind of stays with a temra phenotype for the CD8 cells, Whereas for in the CD4 T cells, you get more of a central memory phenotype. And these, you know, you get less of them, but they're still persisting about, you know, 1% of all your T cells after 6 months. So that's, you know, they don't really disappear, these memory T cells. Okay, so how about the proteins that are targeted by T cells in patients with mild COVID-19 and severe COVID-19? So here you're looking at the T cell specific for the spike protein, membrane protein, and nucleocapsid, pretty much about the same. So here, this is an interesting group here. These are people who were seronegative, who were exposed but didn't develop symptoms. So you get pretty good T cell response in people who've been exposed to infection but were asymptomatic. And these dark blue group, these are blood donors from 2020, and the light blue group are blood donors from 2019. So what is interesting here is you're getting some T cell responses in PBMC from people who have never even met SARS CoV2. And basically you've got quantitatively similar response. So this is kind of interesting. 60% of exposed contacts and 20% of 2020 blood donors in Sweden, they had a SARS CoV2 specific T cell response, and often in people who were seronegative. Okay, what about, so that means, there might be some subclinical infections, and that's enough to give you a T cell response. So what about this group here, people from 2019 who still have got a detectable T cell response? So this has also been, observed by other groups. So this is from Germany here, and they also found some healthy donors who had a detectable T cell response to the spike protein. So then they checked whether it's the S2 domain or the S1 domain. So the people who respond have a T cell response to the S2 domain, not the S1 domain. Then what they wanted to do was they wanted to test the hypothesis that these S2 specific T cells are actually cross-reactive with antigens from the endemic human coronaviruses. So what they did was they stimulated these T cells with peptides from the S2 domain, sorted out the specific T cells, cultured them, and then re-stimulated them with different peptide pools, either from the 229E or OC43 human coronaviruses. So S2 is from, SARS coronavirus 2, SARS-CoV-2, so that they get stimulated. But also you see a pretty good cross-reactivity with the S2 peptides from these seasonal human coronaviruses. So this is pretty good evidence that some people have got cross-reactive memory T cells that are there because people have been infected with human coronaviruses before, and some of these T cells will cross-react with SARS-CoV-2 epitopes. And one of the questions you might wonder about this is that, okay, so, another paper I'm going to talk about now is on the same subject here. So this one has just been published in Nature in the last couple of weeks. So here there's a study of a cohort of healthcare workers in the UK who were heavily exposed to the virus during the first wave. So they're recruited in March 2020, and they're followed intensively for 16 weeks. So at the end of these 16 weeks, they've got blood samples and they've got PBMC. So they've got a group of these healthcare workers who were consistently seronegative. They never had any virus infection detectable by PCR, and they looked at their CTL responses, and particularly against the polymerase and the structural proteins. So the polymerase they're looking at here are, pools of peptides that cover the RNA-dependent RNA polymerase, which is coded by the NSP12 gene, and the helicase NSP13, and this cofactor NSP7. And they compare that with the response against the structural proteins. So here you've got, an interferon gamma ELISpot, and showing people sampled before the pandemic, so there's some background in this. But for those who have been exposed to the virus but were seronegative, they've got a higher response than the pre-pandemic people, but it's just the same as what they was observed in the healthcare workers with a lab-confirmed infection. So these people been exposed to the virus and they've got CTL. So when they compare with structural versus CTL against the RNA transcription complex, that's what RTC is for, so the replication and transcription complex here. What is interesting is that the CTL response against the structural proteins isn't different in people who had a lab-confirmed infection and those who were seronegative, whereas the CTL response against the replication complex here is actually higher in the people who didn't seroconvert. So it's really interesting because it's kind of showing you that, this CTL response here might be protecting against, seroconversion. So why does this happen? They think that these are pre-existing T cells because NSP12 and NSP13, these are pretty much the most conserved proteins in the genome compared to other coronaviruses. And then they looked to find if they could, to see if they could find specific T cell response against these proteins in PBMC samples from before the epidemic. So in 51 people in London, more than half of them have got a detectable T cell response against epitopes in NSP12. So this is from seasonal coronavirus infection, and it cross-reacts with SARS CoV2. So the other thing that they found here is that they also looked at the NSP responses in some of these people in this cohort who had samples, PBMC samples from the baseline at the beginning of the study period and then after 16 weeks. So you could see that in fact, this CTL response against NSP12 is increased, it's getting boosted. So these people have probably been in contact with the virus, even though they didn't seroconvert. And they also have some indication of virus infection because this is an interferon inducible gene, IFI27. And in the people who've got this CTL response, then this is kind of increased. So basically the idea here is that, these pre-existing T cells against the RNA polymerase components of the virus can actually have a protective effect. So this is really important because this might be the basis for kind of like a broadly acting anti-coronavirus vaccine. So these people seem to have come in contact with the virus. They never seroconverted. They didn't have a detectable viral RNA, in their nasopharyngeal samples, but they did have a boosted T cell response. Okay, so one of the questions that's related with this is how about children, okay? Because we know that children are more resistant to, severe coronavirus infection. And we know that children in school are getting more recent seasonal coronavirus infections. So, maybe children have got more of these T cells around and that's why they're more resistant. So this was kind of tested in a paper also from a bit earlier this year, and they had tonsil and PBMC samples from children and adults sampled before the pandemic, okay? So just very briefly here, this is the percentage of positives they found against different viruses and different parts of the different proteins and virus genome. These are the percentage of positives. Basically, what I want you to look at here is ORF1b. So ORF1b is what codes for the virus RNA polymerase and the helicase. And comparing children and adults, there's no difference. Okay, so the pre-existing SARS-CoV-2 specific T cells are not higher in children compared to adults. So this doesn't explain why children are more resistant to the infection. Okay, so moving quickly along now, I'm going to switch to antibodies and humoral immunity. So now some of the first papers that were published here, looked at patients with acute infections over time to find out how the antibodies response develops during the disease duration. So this is days post-symptom onset. So you can see that seroconversion is about 10 days to 2 weeks after the onset of symptoms in people here. Okay, so neutralizing antibodies start, the first one's 4 to 6 days, but mostly, after like 10 days, and the peak is about, 2 weeks. One of the things that's interesting here is that, there are some people who have symptoms, but they don't actually develop any detectable neutralizing antibodies. Now, another thing that's important here is that, generally speaking, people who have a severe infection develop much higher titers of antibodies compared to people who have mild symptoms. Okay, so that is generally true. People who are hospitalized have a much higher antibody response compared to people who are just a little bit sick and then stay at home. So how does the B cell response persist through time? So here you've got antibody titers over time, days post-symptom onset. Red are people with severe infection. Black is people with mild infection. So basically, after the peak, antibody titers are going to go down, okay? And the lines are all parallel here. So if you've got a low titer at your peak, well, after a couple of months, this will become undetectable. So over like 6 months post infection, this is more data from Shane Crotty's lab. I'm getting an echo here. Probably someone's got their mic and their speakers open at the same time, so it'd be good to switch one or both of them off. Probably the mic would be better. So, antibodies titer seems to just diminish gradually over 6 months, but memory B cells, they stay pretty constant. Okay, so here the memory B cells are being stained specifically by fluorescence labeled spike antigens. And so, the memory B cells stay at pretty stable level. So you'll have like antibody titers going down in the serum, but the memory B cells are still there. So it could be easily re-stimulated. Okay, yeah, a little bit more about the duration of these B cell responses. So here they're looking at kind of interesting paper just been published. They're looking at people who got vaccinated but had not been infected versus people who've been infected and then got 2 doses of Pfizer or Moderna mRNA vaccines. So, in the red lines here, these are people who've been infected and then get vaccinated. So if you've been infected and you get vaccinated, you get a massive boost in your antibodies. Okay, if you're not infected, get vaccinated, the amount of antibodies you get is, at least as high as what you get, in fact, even higher than what you get after an infection, because these are the 2 vaccine doses here. So after 2 doses, the people, if you've been vaccinated, you've got much higher antibody titers than if you've been infected. But then afterwards, you've still got this decline over, 6 months. So how about the memory B cells? Once again, over time, these memory B cells, they remain after vaccination. So that's pretty good. So, antibody titers are reduced. You lose easily about 20-fold antibody titer in your blood after over six months after vaccination. But your memory B cells, they're still hanging around at a pretty reasonable number in the peripheral blood. Okay, so I've got about 20 minutes to try and finish up, and I'm going to talk about variants and neutralizing epitopes. Okay, so the message here about the humoral response and memory is, okay, so antibody titers go down, but in fact your memory doesn't entirely disappear, because B cell memory and T cell memory seem to be maintained at a reasonable level, at least over 6 months. Okay, so these new variants, what are they and how do we know about them? Okay, so pretty much for the first time ever, we've had the ability, thanks to next generation sequencing, to really sequence a lot of virus genomes pretty much in real time. And there's been a huge international collaboration to, sequence a lot of virus isolates and identify variants of concern. That is, variants that start out very rare and then seem to become more dominant in the host population, and try and identify them quickly and maybe try and study them because they might be more infectious, more pathogenic, or might resist vaccines. So there are, couple of good websites here that collate all the information and have great online resources. Now, just to give you a little bit of an idea about the workflow of the whole genome sequencing, so what is kind of very convenient is to, you get your RNA sample from a patient and then you'll reverse transcribe it to cDNA, and then you'll set up 2 PCR reactions, one of which has got 110 primer pairs and the other one has 108 primer pairs, and they cover the whole genome. But these primer pairs don't really interfere with each other. So just with 2 multiplex PCR reactions, you cover the whole genome, then you pull them and then ligate Illumina barcodes and send it off for an NGS sequencing run, or Oxford Nanopore or another high throughput sequencing technology. And then you can demultiplex with the barcodes and then reassemble the whole virus genome sequence. And then you send it in to the database, and that will tell you, what variant it is and that sort of thing. Now, one of the problems with this is that each of the consortia that were working on this invented their own kind of nomenclature for the virus strains. So one of the ones that was kind of proposed was this one from Andrew Rambaud, which was to say, okay, we've got to give a name that reflects the phylogeny of the variant. So there are 2 initial lineages, A and B, and then we need to add numbers to the sublineages. So we go B.1.1.7. This was B.1, and then it split, and then the sublineage then split. And then they say, okay, we'll just have 3 sublevels, and then we'll have a kind of specific name. So that is one kind of convention for naming different virus strains. But the other kind of sites didn't really follow this convention. So for GISAID and NextStrain, they used their own kind of conventions. So, for the COVID lineages, pangolin lineages, B.1.1.7, this was called 20I by NextStrain. It was called GRY by GISAID. So at some point, the World Health Organization expert group got involved in all of this and said, okay, what we've got to do is we're going to have simplified the nomenclature. So this is going to be the alpha variant, B.1.351. This was from South Africa. We call this the beta variant. P1, this was emerged in Brazil. This is the gamma variant, B.1.617.2. This emerged in India. This is the Delta variant. Okay, so alpha, beta, gamma, and delta, they're all different from the wild type. And they have all got some particular spike mutations in the receptor binding domain. So alpha has got this N501 mutation, beta and gamma have got N501 plus 2 others, and delta has got a different set of mutations. So now the real worry is, how are these variants affecting neutralization? So maybe these variants escape from the antibody response. And several groups have tested this, and in fact, it's true. So here, these people are testing serum from patients who had been infected with, the first wave SARS CoV2 and had recovered. So they are seropositive, and they test against the wild type spike glycoprotein or with a combination of these mutations. So you can see you've got, you know, reduced antibody titer against the triple mutant. So that was group in Rockefeller, same thing in Oxford, and the same thing in Pasteur Institute of Planas. So here, you know, the titers here aren't so great, and but you know, you can see that you've got some kind of escape from the beta and the delta variants. So B.1.351, that's the beta variant, and compared to the wild type, you lose about a factor of 10 in antibody titer. And these are neutralizing titers, okay? So all of this is testing whether the virus can still infect cells in the presence of antibodies. So there is a significant effect of these variants on neutralization. So how about after vaccination? So here we've got the same studies, but looking at a plasma or serum from people who've been vaccinated. And once again, if you've been vaccinated with the Moderna or the Pfizer vaccines, you've got a significantly lower titer, neutralizing titer against this triple mutation, similar to what you find in the beta and the gamma variants, with alpha significantly lower, but less of an impact. Similar study here from the Oxford group, you know, once again, you lose a factor of about 10, okay, against the beta variant. And similar studies, similar results from the group in Pasteur with working with Olivier Schwartz. One of the things that's interesting here is that, you know, this is one of the first studies to test the Delta variant. So in fact, the Delta variant is not the best at escaping neutralization. Okay, so this is the neutralizing titer in serum for people who've been vaccinated 8 weeks and 16 weeks after vaccination. So, you know, the beta variant is the best at escaping. Delta is significant neutralization escape, but it's not really more than beta. So why is it risen to become like the most dominant variant in the world? Okay, what seems to be happening is that the Delta variant is more efficient at infecting human cells. So here's one study published in Science maybe about a month ago. So they think, or they found that the Delta variant spike glycoprotein has higher fusion activity, and it really stands out compared to all these other variants. Whereas, you know, in Pasteur Institute, they don't really find the same thing. So it's another test of fusion activity. So this is the, you know, wild type virus. Alpha and Delta have got, you know, enhanced fusion activity, but, you know, no real difference between Alpha and Delta here. So maybe it depends a little bit on the system you use. But there are other groups who have found, you know, enhanced infectivity of these Delta variants, particularly due to this mutation, the L452R, which is really only found in the Delta variants. And another group here found that there's better infection in human lung epithelial cells, especially in the cells which have got low expression of the ACE2 receptor. So, you know, Delta seems to be, you know, so there's, you know, 3 or 4 groups of really published data to show that the Delta variant is more efficient at infecting human cells. And it's probably this that has been the motor behind the expansion of the Delta variant, okay, and not really neutralization escape. So, you know, one of the big questions is, you know, do vaccines protect against the Delta variant? So this is data from you know, recently a short report in the New England Journal of Medicine just a couple of weeks ago. So this was just a trial of people who had got a 3rd dose of the Pfizer vaccine. So you've got neutralizing titers against, I think this is the wild type virus and the Delta variant here. People who are after their 2nd dose and after their 3rd dose. So interestingly, there's no neutralization escape detected in this particular study, but you can see that the 3rd dose of the Pfizer vaccine did boost neutralizing activity against the Delta variant in younger people and older people. So probably, existing vaccines and the third booster dose will help to maintain immunity against the Delta variant. And also, very important question, has vaccination really had a protective effect against the Delta variant? So this is also a short paper just been published in the New England Journal. So it comes from kind of surveillance data from Scotland. So it's got a kind of national database that includes a person's vaccination status, sequencing from the viruses that they had if they were infected, and their hospital records and mortality. So like the overall population covers 5 million people, and they were able to follow the results of about 100,000 people infected from April to midway through August 2021, and followed up through to the end of September. So during this time point, they were able to find which of those people had been infected with an alpha variant and which of these people had been infected with a Delta variant, and whether they died or not, basically, whether they'd been vaccinated and whether they died. Okay, so this is the result. So I've kind of simplified the table from this because they had all sorts of categories. They had unvaccinated, partially vaccinated. So I'm just showing you the results of comparisons with unvaccinated to fully vaccinated people. Okay, so so in young people less than 40 years of age, oh no, I didn't put the boxes in. Okay, so young people less than 40 years of age, they had 35,000 infections in unvaccinated people and 17 deaths. So people have been fully vaccinated, only 7,000 infections, no deaths. Now in older people, unvaccinated people, 4,800 infections, 33 deaths. Fully vaccinated people, either with AstraZeneca or Pfizer, about 13,000 infections, 18 deaths. And then in older people, very few people not vaccinated, but still 24 deaths, and several thousand people fully vaccinated, some deaths. So what's the take-home message from this? Well, one of the things that's important here is that they control for differences in the study population, because in fact the vaccinated populations, people who had been fully vaccinated, had more risk cofactors, okay, than the unvaccinated population. So they this crude proportion of people infected who went on to die is not the only factor to take into account. Okay, so there are different cofactors. So the main statistic that they calculated is this thing, the adjusted hazard ratio, that takes into account all the other cofactors. Okay, heart disease, obesity, this kind of stuff. So this number here is the adjusted hazard ratio. So that means 0.12, 0.05, 0.10, 0.13. That means if you've been fully vaccinated, you've got about 10-fold less chance of dying from a Delta virus, SARS CoV2 Delta variant infection compared to someone who has not been vaccinated. So I think this is an important result because, well, firstly, rather scarily, even if you've been fully vaccinated, you can still catch this virus and it still can be fatal to you. Your chances of survival are like 10-fold higher than if you are not vaccinated. Okay, so that's kind of the latest result on whether vaccines protect against the Delta variant. So I was really surprised actually that there are still some deaths amongst fully vaccinated people, but it seems to have been the case. Okay, so I think time's up, so I'm going to skip all of this stuff about neutralizing antibodies. So I'm just going to go straight to the summary. Okay, so for interferons, the things to remember are, there are genetic polymorphisms of quite frequent variants in the population that are related to severity, at least in the European population. Okay, any kind of genetic study, you have to confirm it in a particular population. It's valid for a particular population. So this is true in European population, people with European ancestry. There is a huge impact of rare mutations in the type 1 interferon system and autoantibodies. This accounts for, or it contributes to, like maybe 15% of all very severe life-threatening cases. So that's really very important. In contrast, the cytokine storm, probably only important in a minority of patients, maybe 3 to 4% of severe cases. Okay, T cells. So, there's no obvious defect in the T cell response in patients with severe COVID-19. So it's not like, people are getting a severe infection and dying because they can't produce antiviral T cells. That's not true. So everybody who gets infected and has a symptomatic infection seems to develop a pretty good T cell response. So memory T cells persist up to 6 months. We don't really have more long-term data yet, so that will probably come out in the next few months. What is very interesting is that there is some pre-existing cross-reactive T cell immunity. So people who've been infected with endemic human coronaviruses, some of those T cell responses can cross-react to epitopes in SARS CoV2. And the ones that might be protective seem to be the T cells against the RNA polymerase complex, the RNA replication and transcription complex. So those T cells may be involved in early control of infection, and if you're infected, your T cells eliminate the infected cells and you don't even seroconvert. So that's just some very, very interesting results just been published this month. So for the antibody response, in your serum, antibody titers certainly go down by about a factor of 10 over 6 months after infection or after vaccination. But, memory B cells seem to persist pretty well. The neutralizing antibody response is mostly targeting the receptor binding domain on the spike glycoprotein. That's the bit I didn't show you. But the most potent neutralizing antibodies are really against the RBD, the receptor binding domain. So alpha, beta, gamma, and delta variants all show some neutralizing escape against antibodies generated against the initial first wave virus. So delta, so this is the dominant variant in the world, everywhere in the world right now. Strangely, it's not the most potent escape variant. So beta and gamma show more neutralization escape than the delta variant, but it's got enhanced infectivity. So it's maybe the combination of these two things. So it's got some neutralization escape, but it's got strongly enhanced infectivity. So these two things together mean that this virus variant is really been much more competitive than all the other virus variants circulating in the world up to the beginning of this year. However, importantly, current vaccines still protect against death, even against the Delta variant. So in the future, perhaps we'll have boosters specific for the main variants, but currently booster vaccinations, there's good data that exists to show that you will boost neutralizing antibodies even against the Delta variant with current vaccines. Okay, so I'll stop there then. That's the end of my time. So if anybody has any questions, I'll be happy to answer.